So my research, I basically do engineered adoptive T-cell immunotherapy. So that means that I take T-cells either out of someone's blood or out of the spleen of a mouse, and I engineer them to be able to see and kill cancer more effectively. It's really important for us to develop immunotherapy for ovarian cancer because we don't have any that work yet. Right now we've seen a lot of success with chemotherapies and drugs like PARP inhibitors in certain subsets of patients, but we really need some strategies that are going to work in all the patients that we end up seeing, and immunotherapy actually offers a lot of potential to do that. The way we went about this was basically to figure out what tumor antigens are expressed in many ovarian cancer patients. And we focused on high-grade serous ovarian cancer because that one's incredibly aggressive and it's one that we see in the majority of our patients. And there was actually a really great study done by the NCI several years ago where they characterized tumor antigens that are overexpressed in tumors but lowly expressed in healthy tissue, and mesothelin was one of those. So we developed a T-cell receptor that could target mesothelin and basically made a human version and a mouse version, and that's what we're looking at in this study. In this study, what we found was that the T-cells that we engineered end up migrating beautifully into tumors, but they eventually become dysfunctional. And a lot of work has gone into checkpoint blockade, this idea that you can find a break on a T-cell and block it so that the T-cells are released to exhibit functional efficacy against the tumor. And so we tried one or two different checkpoint antibodies in our study with our T-cells to see if we could reinvigorate them, make them better at killing cancer. And single agents alone and double agent combinations didn't work. We only saw improved anti-tumor efficacy or tumor killing when we used 3 different checkpoint blockade antibodies together. We think in this particular situation, these tumors have lots of mechanisms that are turning T cells off, and each T cell expresses more than one pathway that can be blocked. And so by simply taking the brakes off one of them, there are other mechanisms still at play, so we have to actually overcome more than one of those simultaneously to really release the potential of these T cells. There is a slight concern that we might be exhibiting or seeing on target but off tumor effects. One of the great things about targeting with a T-cell receptor, though, is that we can actually tailor the specific component of mesothelin we're targeting. So, for example, CAR T-cell therapy targets the protein itself. What we're doing with a T-cell receptor is actually targeting a peptide, so just a piece of the protein that's presented in a special presentation molecule. And so if healthy tissue, for example, presents one peptide and cancer presents a different one, if we engineer our T-cell receptor to only see the one that's presented in cancer, then we've actually made our targeting much safer. So that's what we're really hoping to see when we take this into patients. This can be used in other cancers, and that's what makes this really awesome to be working on, is that mesothelin is overexpressed in mesothelioma, so lung cancers, ovarian cancer, pancreatic cancer. So the idea that this strategy can help one subset of patients is not true. It could potentially help many types of patients with many kinds of cancer. I would love to see in the future a situation where a patient could come in and have their blood taken or have a biopsy of their tumor done, and off the shelf we could say, your tumor expresses antigens A, B, and C, and your tumor has obstacles D, E, and F, and so we're going to engineer your cells with the tools that we already have available specifically to get around the issues in your tumor. I would love to see that. It's still years in the future, but in a perfect world, we'd be able to tailor therapy based on a broad tool set that works in lots of people. I see this going to the clinic relatively quickly, actually. The T-cell receptor that we've engineered to target mesothelin is already in processing. It's in the clinical vector processing plant, so it's being made for clinical trials as we speak. And checkpoint blockade is already in the clinic as well. So once Both the antibodies that are used in checkpoint are proved to be safe, and the T-cell receptor itself is proven to be very safe. It's very quick for us to suggest a trial where we put the two together. The implications for cancer therapy for patients using this strategy are huge. Not only do we have one patient subset, we have many. I am incredibly excited to be involved in immunotherapy right now. I feel really fortunate that I got to be involved in immunotherapy now that it's taking off. There are pioneers who spent decades trying to get this to work, and they fought an uphill battle, arguing that synthetic biology could actually be used as a treatment. And now we're really seeing payoff for all of that work that they did. So I feel very honored to be able to work in their field and to get to push it forward into patients even faster.